This is a production of Cornell University.
Hopefully I won't break into a lecture on intro bio. I used to teach in this room with Bob Turgeon. I'm going to tell you today about some recent work that was done mostly by Robin Johnson, who was a former postdoc in our lab. And this is part of a collaborative grant with Sarah Hake and Ann Sylvester, and the bioinformatic data was all analyzed by Chi Sun. So the question was, it was an NSF-funded project, which we're now in a renewal, hopefully, on proximal distal patterning during maize leaf development. And the focus was on how you basically form a boundary between two different developmental fields. So in this case, we're working with the ligule and the auricle, which are separating, in this case, on the maize leaf, the distal blade, which is photosynthetic, of course, and the proximal sheath, which is mostly involved in supporting the plant. And the auricle and the ligule develop together. This is the epidermally derived film, a fringe of tissue, all epidermally derived. And the auricle is derived from L1 and L2 layers. And it forms a nice line that separates these two distinct developmental fields, the blade and the sheath. And they are very much distinct in their differentiation. At adulthood, you can easily tell blade, just from epidermal characteristics, from sheath. They have different functions. They're very different in their structures. So you might ask, okay, my family asked me, who cares about this little blip of tissue on a leaf? It's a film of tissue. Who cares? Well, actually, it's a great system to study how boundaries are formed and how they're organized between very distinct developmental fields. We know from lots of other studies that organ boundaries are often the case where new axes of growth are initiated. So the question is, how do you draw a line on a leaf to separate these fields? What happens there, and what are the mechanisms that specify these distinct proximal distal fates? Okay, so going back to the earliest stages of proximal distal development, it happens first when the leaf is initiated. In this case, there's no ligule. We can't really see any difference between the blade, what will be blade, and will be sheath, which is really just a really bottom portion of this leaf. Primordium fate mapping tells us that clearly. Most of this leaf is now going to be blade. But they're initiated from the sheet apical meristems. This is an SEM I did many years ago of a 14-day-old seedling, ripped off all but two of the leaves. We see two leaf primordia. And like in most other studied organisms, I think everyone that's been studied in plants so far, initiation of leaves in maize is correlated with localization of auxin transport proteins. So this is an image that Robin made from a plant that's expressing YFP-labeled PIN1. PIN1 is an auxin efflux protein, so wherever you see these green lines on the borders of these cells, you can assume pretty much that auxin is leaving that cell. And we can see at the initiation point of the next leaf, which is here, there's accumulation, high accumulation of this PIN auxin efflux protein. And we also can see a line that serves towards the middle of the middle of the meristem, which will eventually form the vascular trace, the midvein. We can see this clearly on these more well-developed P1 and P2 primordia. I'll use those terms a lot. P refers to plasticron. Plasticron is defined as the length of time between successive innervations of a leaf from the meristem. So a P1 leaf is one leaf from the meristem. P3 is three leaves from the meristem, and so on. Plasticron is a length of time. They can last a day or longer. So any P3 leaf from two different plants will not be at the exact same stage of development, but they'll be close. Okay. This just shows that PIN1, we can infer the direction of transport of auxin at the site of leaf initiation by looking at where these PIN proteins are localizing in the epidermis, and we think it's moving down towards where the leaf will initiate. Now the ligule is derived completely from the epidermal layer, and this is basically work that was first described in this detail by Ann Sylvester. Others like Sharman and people before him did a great job describing ligule initiation. I always tell my students, if you want to learn about plant anatomy, maize anatomy, maize morphology, look at Sharman first. But Ann did a great job looking at these early events in some great detail. And she defined that about the P5 stage, so 5 leaves from the meristem, there's this wide band of localized anticlinal cell divisions, meaning they're in the plane of the epidermis. You can see this range of small cells is forming this wide band. We call this the fat band, or the fat line. And later on, a little bit later in that plasticron typically, in a region of the fat line, you can see a thin line that is emerging by basically periclinal divisions, which send new growth outside the plane of the epidermis. And that will become the ligule. And the rest of the development of this is basically elongation of that thin line from this fat line to form a fringe and later on a full-blown ligule. If you look at PIN, this is work that was first described by Sarah Hake in her lab, but Robin did this also in our lab. This is a bright field image, basically of a P5, P6 leaf primordium, probably about P6. We can see this line here, which is the initiating ligule. And if you look at pin localization, we can see very clearly nice stripes of pin localization over the vasculature. That's expected. That's in the L2-derived layer. We can see that here. It's internal. But also there's a striking stripe, a perpendicular pin localization, which will mark the initiating ligule. Very surprising. We can see that in a cross-section here. It's in the epidermis. It doesn't connect with the internal vasculature. So there's some similarities already to ligule initiation and leaf initiation, which is a theme of this talk. Looking at close-up versions of this localization, we really can't infer any one direction of transport. It seems to be emptying from all sides of all cells, for the most part. Don't infer a direction. In terms of ligule mutations, recessive mutations that disrupt the ligule, this is work that was done mostly in the 90s and 80s by Mike Freeling and Sarah Hake's lab, but there are two ones I'll talk about. One is ligulus 1, which is important. This completely removes, this is messed up, removes the ligule and the auricle, but the line is intact. There should be an image there of a close-up of the leaf. Ligulus 2, so in this case, ligulus 1 removes the ligule, but the fat line is there. The boundary is still there. It's a little bit disrupted, but it's made. Ligulus 2 is different. Ligulus 2, there's a little patch of ligule on the margins, but we can see there's actually sheath that is pushed up where the blade should be. So in this case, the fat line is missing. The boundary is disturbed. Not sure why these are showing this. Must be a PC Mac thing. So ligulus 1 and ligulus 2 were both cloned many years ago. They're both different types of transcription factors. That's not important to us right now. But they were cloned in the 90s, but actually Robin was the first person to ever get a ligulus 1 in situ hybridization to work. So wherever you see this purple staining right here, that's ligulus 1 transcript accumulating. Here's the shoot meristem. It's surrounded by lots and lots of these tree-like looking primordia. And we see ligulus 1 first accumulating at about P6. So after the fat line is established, where the thin line is emerging, where this fringe is just starting to grow is where we first really see it. Okay? This is in keeping with the phenotype. The fat line is present, the boundary is there, but the thin line is gone. Ligulus 2 is basically expressed throughout the shoot. This is a big surprise. We don't know what it's doing in the shoot meristem, but expressed everywhere, but it seems to only localize. Okay, so let me speed this up. So there are also a bunch of dominant mutants that disrupt proximal patterning. The most famous one is knotted 1. This is a picture I got from Sarah. It's a beautiful mutation, dominant, gain of function. Most of these, which I'll talk about today, are class 1, knotted-like homeobox, homodomain containing proteins, that's what they encode, that are misexpressed, expressed ectopically in the wrong place or in the wrong developmental time. And classically, what they do is, this is a great example, ligulus 3, another class 1 Knox gene. It shoves sheath tissue distally, so replaces blade with sheath. When this happens, you have a new blade sheath boundary along this axis here, and it's hard to see it here, but every time you have this new abutment of ectopic sheath against blade, we get ectopic ligules forming. So ligules form at the boundaries between blade and sheath. You can't really see it because this is mostly a abaxial view. The ligule is adaxial. But if we were to look at this thing very closely, this is a beautiful phenotype. This is all sheath tissue, which is expressing in the blade, mostly over veins. And if we saw this on the opposite side of the leaf, we'd see a little flap of ligule, every one of those little notches of sheath. So this phenotype led Sarah to specify that perhaps knotted 1 makes sheath. And she described the expression of the RNA and the protein many years ago. It's expressed in the shoot meristem, not in leaves. It's turned off in leaves. We know now that auxin is responsible for turning off Knox expression of RNA in the leaf primordium. And it remains off, the RNA anyway, remains off leaves. It's also in the stem. The protein was shown many years ago by Bill Lucas, Dave Jackson, and Sarah to traffic through plasmodesmata. So we see, as opposed to knotted RNA, which is not in the L1 layer of the meristem, we see it in the L1 layer, the protein in the L1 layer of the meristem. But we also see it in the base of these leaf primordia. We don't see RNA there. So Sarah thinks this is going to be pre-sheath. The sheath is very much unelaborated at this stage of development early on. It elaborates last part of the leaf to grow. So that makes sense. So she thinks that knotted specifies sheath. So what did Robin do? Well, she started with plants and she microdissected them. And I'll show you how she did that. And we did RNA-seq analysis of microdomains, which I'll explain in a bit. She analyzed the data, then we went back to the plants and looked and see where these genes are expressed, and now we want to actually test some of our models, make mutants and double mutants, and see what's going on. So we used laser microdissection a lot in my lab, and what she did was she said, okay, let's go to a P7 leaf where we can clearly see the ligule is just emerging. And she took 3 sections, each one 100 microns tall, pre-ligule or emerging ligule, pre-sheath, and pre-blade. And she left about 50 microns in between to make sure there was no contamination. And this is a close-up of what our laser microdissection scope can do. We target this tissue here. This is a thin section of a shoot apex. It's placed on a slide. These are ugly images. There's no staining. There's no cover slip. We can still see, if we can see it, we can get it. We can see the emerging ligule. And we can blast this now off the slide into a collection tube, make RNA, do RNA-seq. And she did this analysis, only certain reads which map uniquely. The MACE genome is duplicated. If there were unresolved paralogs, we couldn't tell which one it was, we threw it out. Okay, so this is basically how we compared the data. I'm not going to show the gory Venn diagrams, but there are lots of genes which are differentially expressed in these 3-way comparisons of ligule versus sheath, ligule versus blade, and blade versus sheath. I'm going to talk mostly today about the genes which are upregulated in the ligule, and there are a lot of them. So as a control, we looked at ligulus 1. We knew from the in situs and from earlier data that it was expressed highly in the ligule, and this RNA-seq data confirms this, basically not in the sheath or blade, very significantly upregulated in the ligule. So we trusted the data. We think laser microdissection works. So what kind of genes are they? Well, the gene that was the most highly differentially expressed in the ligule, over 108-fold versus the ligule, 50-fold versus the blade, was this gene of unknown function. But it's a grass-specific, so far as we can tell, gene. We don't find putative orthologs of this gene outside the grasses, suggesting that maybe the ligule really is a grass-specific sort of structure. And it's expressed basically a little bit late, P6, right when the fringe is starting to form. And later on, it localizes to this notch, this boundary between the emerging ligule and the blade. And even later on, it's clearly in that notch between the ligule and the blade. A beautiful and powerful expression. But the surprising thing was we also found this gene was expressed at multiple organ boundaries. So this is a close-up of the shoot apical meristem. This is actually a patchwork of several in situ photographs that Robin made. And we see it's expressed at the boundaries of lots of lateral organs between this incipient leaf primordium and the meristem, between this leaf primordium and the meristem or the shoot, between this lateral bud. So it looks like it's expressed at multiple organ boundaries, this grass-specific gene. And this was basically a theme throughout this analysis. And one of the genes we found was CUC2, homologous to a gene in Arabidopsis, which is a member of a lateral organ boundary gene family, expressed at organ boundaries. So it's expressed clearly in this cleft between the emerging ligule in P8 primordia and the blade at the boundary between the ligule and the leaf. But it's also expressed at boundaries between leaf primordia, between lateral bud primordia. It's everywhere. So it looks like ligule genes are boundary genes. Here's another example. This is a gene called BOP4. This is clearly homologous to probably the ortholog, of a gene from barley, which is a unicomb or a branching mutant. They fail to make tillers or branches. They're also ligulous. It's expressed highly in the ligule, shown here, but it's also expressed in these lateral buds. These will give rise to ears in maize, give rise to tillers in barley. So it's a branch primordia gene. We also found two Bell-like homeobox genes which caught our eye. Bell homeobox genes interact with class 1 Knox genes. We know that knotted 1, ligulus 3, these are enough to make a ligule develop. And they putatively interact with these Bell-like genes. So our eye was caught by these two genes, Bell 12 and Bell 14. And again, they're expressed nicely in the ligules, in this notch here, and also in the developing ligule itself, but also in the boundaries around the ligule, not maybe the auricle, that's what that marking, but also lots of other organ boundaries. Ligules and organ boundaries is a theme. Here's another one just to drive this home, Bell 14. It's expressed in a much wider region. It's in the ligule notch, but it's also in the region around the ligule, not localized to the ligule auricle region at all, especially early on. But it's also expressed at multiple lateral organs and their boundaries. So what are they interacting with in a ligule? We know knotted 1 is not expressed in the ligule region, but ligulus 3 is. So we're doing experiments now to see whether or not these bells can physically interact and direct gene expression by binding with ligulus 3 protein. It's expressed higher up in the ligule. Robin showed this. She again was the first person to get a ligulus 3 in situ to work. And it's upregulated in knotted mutant leaves. So this is a good candidate for interacting with bells way above the boundary. This is a big surprise to me. Robin showed me this data and said there's a WOX transcription factor that's the most differentially expressed transcription factor in the ligule. And it's caught my eye because I've been working on a pair of WOX genes for the last 20 years, narrow-sheath 1 and narrow-sheath 2. I asked her, is this narrow-sheath? And actually it is. And I was totally surprised because we described this as a gene which functions in the shoot meristem to specify a leaf domain. So this is a narrow-sheath 1, narrow-sheath 2 mutant, which is missing a big chunk of leaf. And this is made early in the meristem. There's a lot of ligule missing. If you look at the close-up of this blade sheath boundary, there's a lot of ligule missing compared to wild type. But we figured, okay, if you don't make leaf early, you can't make a ligule on that leaf later. So I never expected this thing would be expressed in the ligule. We described it as being expressed in the meristem. This is a cross-section of the shoot apical meristem. This is where narrow sheath is expressed to direct those lateral domains. So we find leaf initiation genes, which are redeployed, reinitiated at the primordium to make a ligule. Here's another example, a gene called fabulosa 1, first described in Arabidopsis as being involved in dorsiventral patterning, adaxial or top layer patterning of leaves. It's expressed in the shoot apical meristem. It's expressed in leaf primordia throughout the vasculature, in the protoxylem and the xylem, which is adaxial. But it's also highly expressed in the ligule. So another leaf initiation gene which is redeployed at the ligule. So the overall theme here is that ligule genes are organ initiation and boundary genes, branch genes and leaf genes, which are redeployed later. And we didn't expect this. It makes perfect sense now, but we didn't expect this. It makes sense because we know that evolution is pretty conservative. It's not really good at making brand new structures. It usually takes and borrows mechanisms and processes from other events and modifies them slightly. So here's basically the model. It's a crazy model. We actually formed this model by drawing all over Sarah Haig's walls in her living room during a meeting we had. And this is the final version. It's complicated, but basically, okay, ligule initiation reiterates lateral organ initiation, but it's not completely reiterated. If you look at, for example, at the localization of pin protein, it connects to the vasculature in a leaf primordium. It doesn't connect to the vasculature in a ligule. These ligules, in maize anyway, are not vascularized. So the idea is that there's a interaction between Knox proteins and auxin. Auxin is repressed by Knox proteins. These boundary genes, like CUX, are repressed by PIN, by auxin. And so NOD is localized to the base of the leaf. we get these stripes of, if you look at it in this transfer section, it's easy to see. Here's pin localization, auxin, which is removing the NOx, and it's also removing the COX. And they form this boundary where this pin is formed, and this is where the ligule will initiate. And we think that these bells interact with some NOx proteins in this region here, which will form the auricle and the ligule. So that's the model. We hope we can get refunded and we can test some of these predictions and test this model. We're sure it's not exactly right. And thanks. I'll take some questions if there are any.
This has been a production of Cornell University, on the web at cornell.edu.